You may think you've never heard of nucleic acids before, but DNA is a specific type of nucleic acid. Another nucleic acid, RNA, is also very important to life, and all nucleic acids are polymers that are composed of nucleotides. There are 3 parts to the nucleotide monomer: a phosphate group, a 5-carbon or pentose sugar, and a base that contains nitrogen, which is sometimes referred to as a nitrogenous base. Here we can see the arrangement of those 3 parts. The pentose sugar is connected to the phosphate group at its 5th carbon, and the nitrogenous base at its 1st carbon. Now, if you remember, phosphate functional groups can bind together by high energy bonds. A special nucleotide called adenosine triphosphate, or ATP, has 3 phosphates bonded together. ATP is used by all organisms to link endergonic and exergonic reactions. But we will look at this molecule in greater detail in another chapter. The pentose sugar in nucleotide monomers can be one of 2 different types. For example, if we compare DNA and RNA nucleotides, we find that the pentose sugar in RNA nucleotides is ribose, while the pentose sugar in DNA nucleotides is deoxyribose. The name deoxyribose makes sense because if we compare the groups attached to the 2nd carbon, we find that deoxyribose lacks an oxygen atom. There are also differences in nucleotides when we examine the nitrogenous bases. Cytosine, abbreviated as C, uracil, and thymine are 3 different types of nitrogenous bases called pyrimidines. They consist of a single ring structure with different functional groups. The base uracil is only found in RNA nucleotides, while thymine is only found in DNA. Please note that while there is no atom specified at the bends in this ring, it is implied that carbon atoms are located in these positions. Bases that have 2 rings fused together are called purines. Adenine and thymine are larger bases than pyrimidines. Please be able to differentiate between purines and pyrimidines for the exam. Now we need to be able to add the nucleotide monomers together to get the nucleic acids. But nucleotides can only join in very specific ways, so let's examine the pentose sugar in a little more detail. Like all carbohydrates, the pentose sugar's carbons in a nucleotide are numbered. As we stated, the 1st carbon is attached to the nitrogenous base, while the 5th is attached to the phosphate group. The apostrophe after the number means prime, so we can call these carbons primes. For example, the 5th carbon is referred to as the 5' carbon. This numbering scheme allows us to determine the orientation of the nucleotide monomer. This is important because nucleotides can only polymerize, that means form polymers, in a certain direction. A nucleotide can only be added to the 3' carbon of another nucleotide. The covalent bond that forms between the 3' carbon of the 1st nucleotide and the phosphate group of the 2nd is specifically called a phosphodiester linkage. As the phosphodiester linkage forms between the pentose sugar of one nucleotide and the phosphate of another, this forms a connected skeleton in the nucleic acid called the sugar-phosphate backbone. And as this diagram shows, nucleic acids are made up of many nucleotide monomers bonded together. One end of the nucleic acid is called the 5' end, as that is the direction the 5' carbon of the pentose sugar is oriented. The other end is called the 3' end. You can also see that the nucleic acid is composed of nucleotides with different nitrogenous bases. As this is an example of an RNA molecule, it contains uracil nucleotides. In addition, RNA is only composed of one strand. DNA, on the other hand, is composed of 2 strands, which run in opposite directions to one another. One strand is flipped upside down in comparison to the other strand. The two strands of nucleotides are held together by hydrogen bonds that form between the nitrogenous bases of the opposite strands, and there are specific rules about which bases can pair up. The base pairing rules of DNA are very specific. Nucleotides with guanine and cytosine can pair up as they form 3 hydrogen bonds. Nucleotides with adenine and thymine can pair up as they form 2 hydrogen bonds. Both RNA and DNA contain information coded in their nucleotide sequences. We will look at how this information is coded in more detail when we discuss genetics.